We just left off with the association areas around the frontal lobes and talking about Phineas Gage and how his personality has shifted, even though he could function as a somewhat regular person, despite these horrible injuries that you see in front of you. But we got cut off before I could talk to you a little bit about some of the other association areas. This part of the video will be much shorter, I promise you. So association areas also perform other mental functions. In the parietal lobes, parts of which were large and usually shaped in Einstein's normal weight brain, they enable mathematical and spatial reasoning. In patients undergoing brain surgery, stimulation of one parietal lobe area produced a feeling of wanting to move an upper limb, the lips, or the tongue, but without any actual movement. With increased stimulation, patients falsely believed they actually had moved. Curiously, when surgeons stimulated a different association area near the motor cortex in the frontal lobe, the patients did move, but had no awareness of doing so. These head-scratching findings suggest that our perception of moving flows not from the movement itself, but rather from our intention and the results that we expect. Yet another association area on the underside of the right temporal lobe enables us to recognize faces. If a stroke or a head injury destroyed the area of your brain, you would still be able to describe facial features and to recognize someone's gender and approximate age, but strangely be unable to identify the person as, say, Lady Gaga or your grandma. Nevertheless, we should be wary of using pictures of brain hotspots to create new phrenology that locates complex functions in precise brain areas. Complex mental functions don't reside at any one place. There's no one spot in a rat's small association cortex that, when damaged, will obliterate its ability to learn or remember a maze. Similarly, the acquisition, development, and the use of language depends on both specialized neural networks and their integration. 19th century research by French physician Paul Broca and German investigator Karl Wernicke led to the discovery of specialized language brain areas. Damage to Broca's area disrupts speaking, while damage to Wernicke's area disrupts understanding. Today's neuroscience has shown that language functions are distributed across the brain areas as well. Memory, language, and attention result from the synchronized activity among distinct brain areas. Ditto for religious experience. Reports of more than 40 distinct brain regions becoming active in different religious states, such as praying and meditating, indicate that there is no simple God spot. The big lesson: our mental experiences arise from coordinated brain activity. Our brains are sculpted not only by our genes, but by our experiences. The last thing we're going to talk about is, really quickly, just brain plasticity. This is kind of interesting, though, to look at, isn't it? Areas of the cortex in mammals and how that's changed over time. MRI scans shows that well-practiced pianists have larger than usual auditory cortex areas that encodes piano sounds. In Unit 9, we'll be focusing more on how experience molds the brain, but for now, we're going to turn to another aspect of the brain, which is called plasticity, its ability to modify itself after damage. Some of the effects of brain damage described earlier can be traced to two hard facts. Number one, severed neurons. Unlike cut skin, usually these do not regenerate. If your spinal cord were severed, you would probably be permanently paralyzed. And then number two, some brain functions seem pre-assigned to specific areas. One newborn who suffered damage to temporal lobe facial recognition areas later remained unable to recognize faces, but There is good news. Some of the brain's neural tissues can reorganize itself in response to damage. Under the surface of our awareness, the brain is constantly changing, building new pathways as it adjusts to our little mishaps and our new experiences. Plasticity may also occur after serious damage. Especially in young children. Constraint-induced therapy aims to rewire brains and improve the dexterity of the brain-damaged child or even an adult stroke victim. By restraining a fully functioning limb, therapists force patients to use the bad hand or leg, gradually reprogramming the brain. One stroke victim, a surgeon in his 50s, was put to work cleaning tables with his good arm and hand restrained. Slowly, the bad arm recovered its skill. As damaged brain functions migrated to other brain regions, he gradually learned to write again and even play tennis. The brain's plasticity is good news for those who are blind or deaf. Blindness or deafness makes unused brain areas available for other uses. If a blind person uses one finger to read Braille, the brain area dedicated to that finger expands as the sense of touch invades the visual cortex that normally helps people see. Plasticity also helps explain why some studies find that deaf people have enhanced peripheral vision. In those people whose native language is sign, the temporal lobe area normally dedicated to hearing waits in vain for stimulation. Finally, it looks for other signals to process, such as those from the visual system. Similar reassignment might occur when disease or damage frees up other brain areas normally dedicated to the specific functions. If a slow-growing left hemisphere tumor disrupts language, the right hemisphere might compensate. If a finger is amputated, the somatosensory cortex that received its input will begin to receive input from the adjacent fingers, which then become more sensitive. Although the brain often attempts self-repair by reorganizing existing tissue, it sometimes attempts to mend itself by producing new brain cells. This process is known as neurogenesis, and it has been found in adult mice, birds, monkeys, and even humans. These baby neurons originate deep in the brain and may then migrate elsewhere and form connections with neighboring neurons. Master stem cells that can develop into any type of brain cell have also been discovered in the human embryo. If a mass produced in a lab and injected into the damaged brain, might neural stem cells turn themselves into replacements for lost brain cells? We might someday be able to rebuild damaged brains, much as we reseed damaged lawns. Maybe new drugs will spur the production of new nerve cells. Stay tuned. Today's biotech companies are hard at work on such possibilities. In the meantime, we can all benefit from the other natural promoters of neurogenesis, such as exercise, sleep, and non-stressful but stimulating environments.